So what stopped SARS, right? So what is this how did we contain this? Because we know COVID wasn't contained, right? COVID got out of the box, ravaged the world, right? So how did they do this? Well, old school, right? Isolate the sick, keep them away. If you're sick and you're not near anybody, you can't give it to anybody, right? Pretty simple. Quarantine, well, you may have been exposed to that person. You need to stay away from everybody as well because how long it takes to develop in you, we need to see, after a certain number of days, depending on what the disease is, then we'll give you the all clear to come out. And quarantining is nothing new. So the word quarantine comes from the word for 40 days in Italian. And back in the 14th century, during the Middle Ages, when big ships would come to port, they imposed a 40-day quarantine. And that's when, through the Middle Ages, diseases would just ravage. And they were scary because we didn't know what diseases were back then, right? We had no idea. And so, smallpox, cholera, the bubonic plague, all these fascinating and deadly and horrifying diseases, they knew enough. Keep them on the boat, and if everybody's all clear it then, let them on land, and we're not going to get a disease from wherever they came from. And then, washing your hands. Washing your hands is always a powerful way to remove the potential spread of these diseases. Face masks, and for all of this to work, you need public messaging. You need the right people out saying you need to wash your hands, right? All this sounds very familiar, doesn't it? So if we then go back to the beginning of this class that I'm talking about, where we followed this through, right? We compared it to SARS. We were looking at the numbers every day. We also looked at the pneumonia and influenza mortality rate in the United States. I do this in all my classes because we have seasonal influenza. A lot of people die in our country from seasonal influenza every year, right? Many millions of people get the flu. And so from an epidemiological standpoint, we download data in my class. We look at the CDC, we look at the World Health Organization. So if we look at this image, we have three lines. We've got two black lines and we've got a red line. The bottom black line, right? And first, if we just kind of look at this, right, what we see is up, down, up, down, up, down. It's a seasonal influenza. And so what we see is in the winter, we have the up, and in the summer, we've got the down. It never goes away. It's still circulating, which is why it can come back pretty regularly in the fall and the winter. So what we're seeing is, for example, here in this particular year, the red line doesn't really go above that top black line much at all, right? And so what we're seeing is not much of an epidemic there. And this following year, okay, a little bit worse of a flu season. And what we're looking at with the red line and all these numbers is the percent of people that died in the United States, total people, that died of pneumonia and influenza, right? And so influenza is underreported on death certificates, but a lot of people will die from pneumonia. Influenza is a contributing factor to people, congestive heart failure and other diseases. And so basically, a way to track respiratory diseases is a way to look at death records and did you die from pneumonia or an influenza? Put the numbers together. What is the percentage of those deaths out of all of the deaths in the United States? And that's what we're looking at here. So what we see then is that 2017 and 2018, that was a bad flu year, right? Not only did we have a high number of cases, but we had a high percentage of all deaths in the United States from pneumonia and influenza. And then we had a quiet year. And it's not unusual for there to be these years where we have really, really bad flu seasons and then some years where it still kills people, kills thousands of people, so it's not a good thing. But then here we were at the beginning of this class, right? So in the beginning of January of 2020, this is the numbers we were looking at. So it looked like, well, the flu season, maybe it's going to be mild. Maybe we're not going to have many deaths from pneumonia and influenza. So we track this every week, but if we just go ahead and fast forward to how this looked at the beginning of July, that's what the curve looked like. So look at the percentage of deaths. Look at that massive spike, that massive red spike going up to about 16% of all the deaths from pneumonia and influenza, right? And so not just from the flu actually had a truncated version because everybody was sheltering in place, staying away from each other. Some people were wearing masks, and so it truncated it. But the number of deaths from pneumonia, of course, increased due to COVID. And we know that there's a lot more deaths than just people that died of pneumonia. We're having different types of organ failure. This is just an example of how a respiratory disease in a particular year where people die of pneumonia, it all gets lumped together so we can see the disease burden of respiratory diseases. And so the burden in the United States is, and that's what we call it. We call it a disease burden or the flu burden. And so when we're thinking about this, so what we've done is we've talked about the very first report. We've talked about how my class has followed it, followed the numbers, followed the news reports, learning it is human-to-human transfer. The cruise ships, right, that were just incubators, absolutely horrifying. And so, when we look at this, how do we kill COVID, right? And why can we kill it with these particular measures? Well, outside of the body, it's easy. So when we look at this particular image, there's the red spikes, which is what allows it to bind to your cells and get in. But then all the gray ball, that right there is the lipid bilayer, and that actually came from somebody's cells. So when the virus is packaged inside the human cell, when it comes out, it wraps itself in a human cell membrane, right? And that's what it has on it. And since it's a cell membrane, it's essentially made of, we can think of it as like fats. So just like detergent cuts through grease, detergents, soaps, alcohol will cut through the sphere of this and destroy it. So it's easy. Bleach just kills it. Lysol, not a problem, right? And however, you're not going to start ingesting these to get rid of COVID. They won't go to the right place. And Lysol and bleach have no business in or on your body, right? So what do we do for inside the body? Well, inside the body, it's a whole other story, right? And so that's where right now what we're going to do is we're going to take a hard right into a little bit of medicinal chemistry. And so as a biochemist, when I'm at a higher level, we would go into the structures of these in detail. We talk about actually how it binds to particular enzymes. For a lower level microbiology class, I treat it different. We just get to the nuts and bolts. What do we need to know to understand how it affects the disease? So that's what we're going to do right now. And so if we look at these two molecules, I still, even this many years out, I love looking at the structures of these organic molecules. It's just fascinating. My mind, they're twirling around in my mind. I'm looking at how they'll react to things. But let me ask you this. At the beginning of COVID, what are some of the drugs that made the national headlines over and over again in clinical trials? Some people really pushing them. Can you think of two of them? Hydroxychloroquine, absolutely. That was one of the ones that made a lot of national attention, right? We're in a lot of trials, even early on. How about the other one? One, a broad antiviral from the company Gilead. It's a drug that had been known to be effective to some degree against SARS, against Ebola, Nipah virus, so a broad spectrum antiviral. Anybody? Remdesivir. And so that's what these two structures are. One is remdesivir and one is hydroxychloroquine. So what we're going to do is we're going to take just a little bit of time to look at how this virus infects a cell and how these drugs work to prevent viral replication. So first of all, this lower part here, this gray, right, is the human cell. And so this is the surface of the cell, and if we go all the way over here, what we see is this ACE2 receptor. And so this ACE2 receptor is where the spike protein, that little spike right there, binds to. And so once it does that, it latches on and it tricks the cell into bringing it in. And so that's what this membrane fusion is. And so now it's inside the cell, but it's in this endosome. It's been packaged up and it needs to get out. So it triggers a way to get out, and it's called uncoating. And so when it uncoats, it shoots its RNA genome into the cell. So if you're a cell that's going to make more viruses, the genome needs to be replicated, right, the viral genome, and you need to make the spike protein. You need to make all the proteins that are in there. So that's what's going on all this, and this is a whole lecture worth, and we're not going to get into that. But basically, over here what we have is, down here we've got the proteins being made, and they're being assembled here. And then once the RNA genome is synthesized, it is stuffed in there as well. It's assembled, and then what happens is after you have a virus assembled, it then leaves the cell. And so while this shows one virus going in and one virus going out, more than one virus can infect the same cell. And you don't just get one virus coming out, you get thousands of viruses coming out. It gets to the point where the cell just is only making viruses that it dies. And that's how you can get lung tissue, for example, in the lungs. That's how it can be damaged. And so everywhere that I have yellow are potential places to try to block. So let's block some of these stages, and if we can block any of these stages 100%, then we can prevent this cell from making more viruses and infecting it. And before I forget, vaccine. So a lot of vaccines are actually targeting that spike protein that's on the outside. And so remdesivir blocks this RNA synthesis. It blocks the genome from being replicated. Now what happens is when the virus comes in, it comes with its own enzyme, which is encoded for in its genome. So when your cell makes more viruses, it's making more of that. But it comes with its own enzyme. And what it does is it starts making the genome. And so this particular enzyme, remdesivir, blocks. And the way it does this is, I think, genius. These medicinal chemists, I tell you, very impressive. I love it. So what we're looking at is on the top remdesivir and on the bottom ATP, so adenosine triphosphate. And so when you need to make an RNA genome, you need to take the A's, the U's, the C's, and the G's, all the individual nucleotide triphosphates, and then the viral enzyme will add them in the right order together to make that long chain of RNA genome, and then it can be packaged. Okay, so let's look at some similarities here. So if you look at the top remdesivir, how is it similar and how is it different to ATP? Because to make an RNA genome, you need to put this in from time to time. So if you get remdesivir inside the cell, then sometimes it'll grab that instead. Instead of putting in an A, it puts in remdesivir. So how does that prevent viral replication? Well, look at the two structures. What areas look different to you and what look the same? And what pops out? And so yeah, so that's a big difference right there. Right? So if you look at this particular area compared to down here, this triphosphate, this multiple phosphates can't get into the cell very well because the external part of the cell is negatively charged, so it will repel it. So scientists said, hey, let's put this thing on there. It can get inside the cell, and by the way, once it's inside the cell, there's enzymes that will cleave that on and add 2 more phosphates so that it's then got the phosphate group. All right, so the next difference that I'll mention is this cyano group. And so this ring in here, this ring right here, it has a particular shape, okay? And so when it has that cyano group, it causes the shape to change and it puckers it. So it kind of changes the shape. And once it changes the shape, well, if the enzyme brings that in and it's got that puckered shape from the cyano group, it's going to get jammed. So the enzyme can't work anymore, and you stop viral replication just like that. And so the last feature is this carbon-to-carbon bond up here. And what that does is normally there are enzymes which can cleave this, right, this bond right here, and so this makes it more stable. So it can get in the cell, it's more stable, and it jams that RNA-dependent RNA polymerase, and so you cannot create more of the viral genome. Love it. And so that's what remdesivir does. That's how it blocks RNA synthesis, and so then you cannot get the rest of this process going. And now hydroxychloroquine, we're not going to get into all of it, but if you look at all the X's around this particular image, what you see is that we believe we have evidence that it does block different stages, and that's really nice to be able to block at different efficiencies, different stages of this infection. And so, you know, we've talked about SARS, we've talked about COVID, we talked a little bit about influenza, not much. And so when we talk about these diseases, what else is out there? What other type of viruses do we have out there? Well, there's more. There's a lot more. And so if we just look at a handful of viruses that I'm used to teaching about, some are very common in this country, others are not. Some infect humans, others infect plants and animals. You know, as part of how I teach, for example, when there was the Zika outbreak in South America that made its way up to Florida a little bit, we covered that. Every year there's West Nile fever going through the United States. And when the Ebola outbreak was happening, and we got some here in the United States, we covered that as well. And so, however, what are scientists really worried about? What could be the next major pandemic? Now at the time of taping this, COVID is ravaging us. But that's not what we were necessarily predicting, although some scientists were saying SARS could be the next big one, and this is SARS-CoV-2. But really, the one that's been scary for scientists and public health officials for a long time is influenza, specifically bird flu. And so influenza is really a crazy, interesting virus where, for example, pigs, they're great incubators. They can get infected with a human influenza, a bird influenza, and a pig influenza all at the same time. That can get all mixed up, and what comes out is a mixture of all of them. It's like a Frankenstein influenza that your body's never seen before, right? And so this is how we get pandemics with influenza. Now, bird flu is particularly scary because bird flu has a 50% case fatality rate. Imagine if COVID-19, easy to spread, 50% of the people that got it died. Horrifying, right? So the thing that the silver lining with bird flu is that it's not, human-to-human transmission is exceedingly rare, exceedingly. So unfortunately, when some people get it, a lot of them die, but at least they're not then transmitting it to other people. Because that, we just can't have something like that. And so whether it is influenza, whether it is from bird flu, whether it is Ebola, whether it is COVID or SARS, a lot of these diseases jump the species barrier because of unnatural conditions we put them in high numbers. And so, when it comes to thinking about this, the COVID pandemic, we expected pandemics to come, and this article from Time Magazine, for example, was published 3 years before COVID, and it has a lot of good information of why we're not ready for the next pandemic. And if you just look at some of the text here, right, it'd be world changing, and COVID-19 has changed the world, no doubt about that. We don't have a vaccine for it. With the drugs, we started throwing at it. Some are working, some are not. And this is particularly interesting because when people get pneumonia, up to 25% of the cases, we never figure out what gave it to them. Gave it to people. So, and pneumonia can be caused by viruses, bacteria, and fungi, and very rare parasites. And so, these excerpts from this Time article, the U.S. healthcare system is not ready for the stresses of a major pandemic. I think we saw that in some major cities. I know we saw that in COVID, right? And then, even talking about mechanical ventilators, which has been a huge concern, right? And so, there's a lot of reading out there. Part of my interest, I love chemistry, I love microbiology, I love it all, but the historical context of how diseases have really influenced and changed humanity is remarkable. It's for example, just as one quick aside, the Great Potato Famine was from a fungal infection to potatoes, and that caused a massive diaspora of a lot of Irish people coming to the United States. So even migrations of peoples have been driven by infections, and that was an infection of a plant. And so, if you're interested in this type of information, I would encourage you to read this book right here. So I use this every year with my students. It's easy to read. It's for the general public. It's fascinating. A great writer. And the very first introduction is about SARS, and the last paragraph is about SARS and a little bit about HIV. And it talks about things basically predicting COVID-19 in terms of global economic massive problems. It even quotes Dr. Anthony Fauci because he's been the head of the National Institutes of Allergy and Infectious Diseases for decades. So again, for further reading, I give my big thumbs up to this book, Deadly Companions.